name is Karina Panucia Tillan. I'm a PhD student from the ICT School in Computer Engineering and Science, and the title of the research is Information Integration for Biological Data Sources, Combinatorial Optimization, and Mathematical Programming Methods. Here we can see an overview of the main issues that I'm going to speak about in this presentation, and I'm going to start with the brief description of the research area. The research area is focused on the development of efficient and effective computational tools for the discovery of transcription factor binding sites in non-coding DNA. To be able to understand the problem, it's important to know what is a transcription factor binding site. There are key elements participating in the transcription process of genes. This process is activated when regulatory proteins bind to specific DNA sequences known as transcription factor binding site, giving place to the starting of the expression of genes. Basically, transcription factor binding sites are provided by experimental techniques, but unfortunately, these techniques are time-consuming and do not scale up well with respect to the large quantity of available data. With the aim to help biologists in this research, we have that since the beginning of the bioinformatics, these problems have been formulated as an algorithmic problem using mathematical models and based on statistic and probabilistic techniques. In these fields, we have that transcription factor binding sites are going to be modeled as DNA motifs. What are DNA motifs? Well, they are short and recurrent patterns of DNA sequences that are presumed to have a biological function. And the problem related with its research is the motif discovery problem, where given a set of unaligned DNA sequences that are thought to be correlated by the same transcription factor, the idea is to identify motifs of a fixed length that are going to represent a motif model of the transcription factor binding site of interest. During this year, one of the activities carried out related with this problem was the development of a Motif Finder, in this case, named Gapped Motif Finder, GMF. And in the design of this problem, we found that the research or the identification of transcription factor binding sites is a challenging task, mainly because transcription factor binding sites are characterized by a high variability in their inside, and the place where transcription factor binds may be located quite far from the transcription start site. During the last 30 years, we have that several methods have been developed to give solution to this problem. Among them, we can find meme, alignase, space, with the bioprospector, and multi-sample, among others. And the main techniques used in their implementation are basically enumerative and statistics techniques. But despite the number of existing methods and the techniques employed, we have that the prediction accuracy of these tools of motif prediction is still very low. Here we propose a new method, the GMF, and I'm going to explain the algorithm of it. Basically, there are three steps: core identification, a core extension, and the scoring of motifs. The core identification we are going to use with the scoring function, and we are going to allow in the structure of the cores the presence of a conservative gap. Later, these cores are going to be extended considering the most promising flagging base, and finally we're going to score the motif. Now I'm going to briefly explain each of these steps, but first I would like to remember that the motif discovery problem receives as an input a set of unaligned sequence, a set of unaligned DNA sequence and a motif length with the aim to identify the CI instances or overrepresented patterns that are going to model the motif. Basically, for the first phase, we have that the core identification, we need as an input the core length of the core that we're going to search, the number of gaps allowed to be inside the core, and the quorum of presence in sequence of these transcription factors or the motifs by itself. The core LS, the length of the core, is composed by the conservative position and the number of gaps that we are going to allow inside of the core. The phase will start by making a scanning of all the input sequences, looking for patterns motifs with G gap and C conservative position. Later, these instances of this core must satisfy the quorum Q of presence in the input sequences, and later we are going to check if the motif core are not highly degenerated, and finally we're going to score the obtained core motif using the width and scoring function to only retain those cores that are most promising. Later, the next phase will be the core extension. Here, we have as an input the core list obtained in the previous phase. We keep the quorum of present on sequences, and we have the motif length to be finally searched. The idea here is, when we have the core of motifs, the idea is to make an extension until the maximum length that we want to search for a motif. We do this extension making all the possible combinations to the left and to the right, and every time we do these extensions, we evaluate, we assess the final motif to keep in the final list those motifs extended with higher score. Finally, we have a big list, and from this list, we are going to return the top scoring motif. For the scoring function used to evaluate motifs and using inside the method, we use the width scoring function to score the core motif, and we use the information content applying also or including also the dependent position. In this case, this is the formula of information content, and in this case, the dependent information among size or position inside the motifs, we are going to use the young information content. These two functions are going to be combined to score motifs in this way. We are going to apply the information content to evaluate the core of the motif and the young information content to evaluate or to assist the flanking bases. Later, the method has been assessed with a benchmark, that is a Thompson benchmark. And here we compare our method against some of the most noticed methods developed in this field. Here, the Thompson benchmark consists of the data set of 4 different organisms. And we can see how our methods for 2 of these organisms overperform all the existing ensemble. And for those organisms where our results are not better, we can see that it remains at the level of the well-recognized tools. Another of the activity carried out was the construction of an ensemble method. In this case, ensemble methods emerge as a solution to improve the accuracy of the single motif finder, we can say to improve the prediction obtained by single motif finder. In this case, ensemble methods include in their inside the execution of different ensemble methods to finally obtain the results obtained by each of the execution of these methods, and later analyze it by learning techniques and give final a result. Even if it's true that ensemble methods overperform the prediction of single motif, it's not clear which are the combinations that give us the better solution. But unfortunately, as ensemble method fits the motif finder, fits the learning rules, and modification requires knowledge of the programming language, make a deeper research on this direction is a little hard and a little difficult. That's why we here propose a general ensemble architecture where we can easily extend the ensemble, including new motif finder and new promising ensemble, and it will be also easy to customize. Here, the general ensemble, instead of implementing or instead of design each wrapper for each external motif finder, we are going to use a general wrapper that is going to interact with the external motif finder through XML configuration files. Later, we are going to obtain these results, and we are going to pass as ensemble methods to learning functions. That is also a module that we have done in a way that could be easy extendable. So if we create new and promising learning function, could be easily included also in the tool. In the prototype that we implement, named CA3 Customizable and Easily Extensible Ensemble, we give the possibility to easily include new motif finder. Here, the general wrapper main functions are the execution of external motif finder and to parse the output obtained for this finder. These main two activities, or these two main functions, are going to be able to execute by the proper definition of two XML files. One of the files will be related with the execution of external Motif Finder, and the other one with the parts of the output of Motif Finder. Fortunately, all the existing Motif Finder, or almost all the existing Finder, the structure of the execution and the parameters are more or less similar. So this gives us the possibility to generalize in some way an XML file to be able to configure the way in which these Motif Finder are going to execute. And the same things happen with the time when we have to define the parts to output the results of the motifs. Here is an example. We can see an example of how can we define the parts file from an output of one of these tools. Here we identify the main information that are of interest when we identify a motif, and they are easily described inside this XML file. Later, as I also say, new learning functions can be easily included inside Ensemble. This is done by the definition of a general Python interface and by the definition of a configuration file also in XML. In this case, the XML file is going to be used in case the new learning function needs of specific parameters, and also to know that there is a system of a new learning function that we can choose, and the general Python interface to be able to communicate with the external learning function. In this case, new learning functions can be implemented in different languages of the Ensemble. Finally, another of the tasks performed or the activities carried out during this year was the construction of the composite Motif Finder. We found that in metazoans organisms, the expression of gene or the transcription process could be carried out or could be activated with the co-occurrence of transcription factors. This suggests that we have to search on a line sequence composite transcription factor binding sites, which means that this is a hard combinatorial problem to face. In this case, we propose an algorithm based on combinatorial techniques that is divided in 4 steps. In this case, motifs are given as input using the position weight matrices representation, and the idea is to identify different transcriptions that add together in the transcription process. For this case, we need to properly separate the classes of transcription factor to be able to search later. In this case, we first compute the similarity among matrices using an utility of the RSAT tool, and later we cluster this motif using the single linkage or the single linkage followed by dense score computation. Later, as this representation of motifs is so generalized, we should identify the positions inside the sequences. Inside the sequences to be able to later search for the combinatorial of the groups that are going to be extending to what we know as the cheese regulatory modules, that are going to be the pairs of co-occurrence of transcription factor binding site, to finally eliminate weak groups and cluster remaining groups considered matched with. These are the experiments performed. We compare the tool with 9 state-of-the-art tools developed for the composite Motif Finder. In this case, we can see that in almost all the values we overperform, we obtain higher value against the existing algorithms. In this case, we use noise zero, which means that the input of the tool are motifs that are present in all the sequences, and later we include some noise which will difficult the identification, and also in this case we can see that compared with the name of state-of-the-art of the tools, we also obtain with good results. This is also an assessment that we have done against one of the most notable tools in this field, and here also we overperform the values. And as a conclusion, we can say that we can see that in the solving of the multi-discovery problem, the use of combinatorial methods can give us a good result. The combinatorial methods have to be considered in the design of a new methods, which means the need of a deep study of which could be the proper combination to be used in the resolution of interest problem. We make a proposal of a general ensemble architecture where new motif finders and new learning functions can be easily included, and the features of CE3, the ensemble, makes the presented ensemble a suitable tool to perform experiments in the search of a proper combination of tools and learning functions. Publications, and I think that's all. Questions?
Okay, thanks for your talk. What do you consider your most significant result? I'm not an expert in this area. I am very far from this.
Well, this area is really difficult. Until now, I will say that the most promising result is the last one, the GMF, followed by the ensemble method.
Is it a very new area, or is it something which is going on from, say, 3-4 years?
Yes, it's something that has been developed since almost 30 years. It started everything. But as you can see, I will say that the strength of the methods start in 2000, more or less. That is the place where we can find the publications more notable. But as we can say, the prediction accuracy even of those tools, even now, are very low. So it's not easy to obtain. So it's hard. It's difficult.
Okay. Because you have to do with the complicated optimization problems, which are hard, intrinsically hard, and you need simplified techniques to reduce the computational load. That's the problem. Okay, thanks again.
You're welcome.
Okay, the final talk of the first part will be given by Silvia Rota.